Good day, everyone. For today's session, we're going to discuss about hemoglobin variants. Hemoglobin variants are different hemoglobin types that are not of the normal form. So let's start. Okay, so I think you have encountered this photo before. So it means the primary function of our red blood cells is to carry oxygen from the lungs and distribute them to the cells of the body. So, what are these hemoglobin variants? They are modified forms of hemoglobin and they can be acquired, and the structure of this are modified by drugs or environmental chemicals. So, the three modified hemoglobins that will be discussed in this session are the following: methemoglobin, sulf hemoglobin, and carboxy hemoglobin. So methemoglobin is a form of hemoglobin that contains iron in ferric state. So ferric state is actually oxidized form of iron. And your ferrous state is the one that is in reduced form. So methemoglobin is also known as hemoglobin or HI. Continuously, it is formed within our red blood cells in small amounts due to spontaneous oxidation resulting from overload of oxidant stress owing to ingestion of strong oxidant drugs. So when your hemoglobin is being oxidized, unlike a normal hemoglobin, it cannot bind oxygen very well. So this is prevented from accumulating within the RBC by reducing oxidized heme by several enzymes systems. Okay, that's the enzyme pathway called methemoglobin reductase pathway. By the way, methemoglobin reductase is the enzyme that allows our ferric form of iron to be reduced back to ferrous form. So through that enzyme, okay, the methemoglobin reductase, okay, or the other name for this is diaphrase enzyme. Okay, this is actually the one and the same enzyme that reduces your ferric form of iron into ferrous form of iron. So it maintains the normal concentration. This enzyme maintains the normal concentration of methemoglobin within RBC. Okay, so of course we cannot avoid the oxidants around us. So again, they can be taken through some drugs and they can be available in the environment. So that's why we cannot avoid being exposed to it. So the main role of diaphrase is to maintain the normal concentration of methemoglobin within our RBCs in less than 1%. So again, in this picture, it shows that ferric is the oxidized form, and it will be reduced back to the ferrous form through this enzyme called NADH methemoglobin reductase, or in short, methemoglobin reductase, or the other term is diaphrase enzyme. So the color of this kind of hemoglobin is brownish to bluish in color. And this particular hemoglobin does not revert back to red on exposure to oxygen. So when one or more iron atoms have been oxidized, hemoglobin molecule changes. So in line with the changes, okay, so what are the changes that will happen, when it is, or one or more iron atoms have been oxidized. Number one change is the oxygen affinity of the remaining heme groups increases. So that will not allow the release of oxygen, and also there will be shift to the left in the oxygen dissociation curve, and oxygen is not delivered efficiently to the tissue. So those are actually the hemoglobin molecule changes that will occur once one or more iron atoms have been oxidized. So when your methemoglobin levels reaches greater than 30% of your total hemoglobin, that can lead to cyanosis or hypoxia. Okay, so this is an example of your normal hemoglobin where each heme molecules are um, attached with ferrous form of iron. And this one, as you may see, okay, there are two ferric atoms attached to two heme molecules in the hemoglobin. So this one is an example of methemoglobin. That one and this one. Now, elevated levels of methemoglobin may be seen in the following. First, presence of oxidants. Second, decreased activity of methemoglobin reductase due to genetic deficiency. It could be NADH, methemoglobin reductase enzyme deficiency, or diaphragmase enzyme deficiency. And the last one, it could be seen in inherited hemoglobin M disease. Okay, so by the way, of course, when there are a lot of oxidants, okay, in our environment, for example, okay, we will be succumb to these oxidants, and they will um, oxidize our ferrous form of iron into a ferric form of iron. Thus, your hemoglobin, normal hemoglobin, will be changed to methemoglobin. So as I've mentioned, the enzyme, the diaphragmase enzyme or the methemoglobin reductase enzyme, this particular enzyme is within our body, and the work or the function of this particular enzyme is to reduce any ferric form of iron into ferrous form. But what if there is a decreased activity of that particular enzyme due to genetic deficiency or um, uh, defective enzyme function? So of course, a lot of ferric form will not be reduced back to ferrous. Okay, and then when the patient has this particular inherited disease called inherited hemoglobin M disease. Okay, by the way, this is caused by a variety of mutations in the alpha and beta chains, which results in the production of methemoglobin. So there is changes or mutation in the genes of the patient's alpha and beta chains. That's why genetically, okay, the patient have or has this particular disease called hemoglobin M disease. So for the treatment, by the way, methemoglobinemia could be acquired or could be congenital. So for acquired, the removal of offending substance could be one of the treatments. So how do we remove? Like for example, if the offending substance or the oxidant is coming from a drug, so the patient should remove that particular drug from being taken from the body. And then administration of ascorbic acid or methylene blue can be an effective treatment. And then the use of oxygen inhalation techniques could also be one of the treatments. So how do we diagnose? So through a peripheral blood smear, we can identify that a patient may have methemoglobinemia when there are presence of Heinz bodies or denatured hemoglobin in his or her peripheral blood film. Also, we can screen the patient with this kind of test, the diaphoresis enzyme screening test, in order for us to know whether the enzyme, the diaphoresis enzyme, in the body of the patient is not deficient. And then we can also do methemoglobin quantitation by knowing the levels of our methemoglobin in the body, and this can be detected by spectrum absorption and instrument. And the particular hemoglobin, this methemoglobin um, variation of hemoglobin can peak in the range of 620 to 640 nanometer at a pH of 7.1. So that's how they can identify and quantitate methemoglobin through spectrum absorption instruments. So what is Heinz body, or what are Heinz bodies? So in the picture you have their Heinz bodies. It is an RBC with inclusions, your Heinz bodies inclusions. They are actually denatured hemoglobin. They are usually found in the cytoplasm area of your RBC, and they are considered abnormal. Thus, when these Heinz bodies, okay, are detected by macrophages, they will be eaten or phagocytosed by macrophages. Okay, so this particular RBC with Heinz bodies, okay, cannot live longer, cannot survive until 120 days. Hence, they have decreased RBC survival. Now let's move on to the next hemoglobin type, or variant hemoglobin, your sulf hemoglobin. Sulf hemoglobin is actually modified chemically, and they are formed by irreversible oxidation of hemoglobin by certain drugs such as your sulfonamides, phenacetin, acetanilide, or phenazopyridine. So your iron here is in ferrous form, okay? But its affinity to oxygen is slower than normal hemoglobin. So outside the body or in vitro, okay, it is formed by adding or by the addition of hydrogen sulfide to hemoglobin and has a greenish pigment. So the characteristic of this kind of modified hemoglobin is that it is ineffective for oxygen transport, which is 100 times less affinity, okay, for oxygen. And it cannot be converted back to normal hemoglobin like your methemoglobin, right? So methemoglobin can be reverted back to the normal hemoglobin through the enzyme, but this one, the sulfhemoglobin, cannot be converted, okay, back to the normal hemoglobin. So once your hemoglobin, okay, has already been modified into a sulfhemoglobin type, then it will persist for the life of the cell until RBCs are removed from the circulation. Okay, so how do we identify sulfhemoglobin? It is again through a spectral absorption instrument, and it peaks at 620 nanometer. Next, modified hemoglobin is your carboxy hemoglobin. It is also known as carbohemoglobin, and the oxygen molecule bound to heme are replaced with a carbon monoxide. So instead of oxygen molecule, okay, that is attached to the heme, okay, carbon monoxide or the CO will attach to your heme of your hemoglobin. So it is caused by binding of carbon monoxide to the heme iron, and hemoglobin has about 200 to 240 times more affinity to carbon monoxide compared to oxygen. So in that sense, when you're, for example, when you are exposed to carbon monoxide, okay, although hemoglobin can normally attach to oxygen, when there is exposure to carbon monoxide, the affinity of carbon monoxide compared to oxygen to your hemoglobin is greater. Okay, imagine it is 200 to 240 times more affinity, okay, to carbon monoxide compared to oxygen. So carbon monoxide combines with hemoglobin slower, but when they are already together, it binds firmer compared to oxygen and your heme iron combination. Okay, so in that sense, um, although carbon monoxide can bind to hemoglobin slower, but when they are binded together already, they are firmer, okay, and they cannot be easily detached, okay, like your oxygen. So since it is firmly attached to your hemoglobin, your carbon monoxide, it is firmly attached to your hemoglobin, it is the release of carbon monoxide is 10 times slower than the release of oxygen. So imagine when your carbon monoxide it has already attached to your heme iron, so it will be harder for the body to release carbon monoxide from the system. Okay, so it is said here that release of carbon monoxide is 10,000 times slower than the release of oxygen. So carboxyhemoglobin is termed as the silent killer because this particular gas is odorless and colorless. And the patients, when exposed to carboxyhemoglobin, can quickly become hypoxic. So like the other modified hemoglobins, this kind of hemoglobin can also be detected through spectral absorption instrument at 540 nanometer. So the source of our carbon monoxide can be endogenous, okay, meaning inside internal factors, or exogenous from the outside factors or environment. Okay, so what are the exogenous sources that are usually available in the environment? One is the exhaustion of our automobiles and from the industrial pollutants such as coal, gas, charcoal burning, and tobacco smoke. So what are the toxic effects of carboxyhemoglobin? We have headaches. The patient can feel headaches, dizziness, coma, and convulsion. This gives blood a cherry red color, which also imparted to the skin of carbon monoxide poisoning patients. So how do we treat a patient with carboxyhemoglobin or being poisoned with carbon monoxide? Patients being poisoned by carbon monoxide, they can be treated with administration of hyperbaric oxygen. So laboratory screening test for carboxyhemoglobin is done through this particular procedure. First, the whole blood is being hemolyzed. The sample should be 0.5 ml of whole blood, hemolyzed whole blood with distilled water, and add 1 ml of NaOH, 1 mole per liter. So more than 20% of carboxyhemoglobin can appear red, okay, or light cherry red. Okay, but if the blood is normal, there's no carboxyhemoglobin, it will turn to brown. Okay, so that's how you identify um, that your sample or the patient has been poisoned with carboxyhemoglobin through that particular procedure. So in this picture right now, you can see that this normal hemoglobin here, okay, can bind oxygen, and yes, can also bind carbon dioxide. But um, with this particular hemoglobin here, oxygen and carbon dioxide can no longer be carried because carbon monoxide binds very tightly to hemoglobin. Okay, so of course we know that our hemoglobin, normal hemoglobin, can carry oxygen and can also carry carbon dioxide at the same time. Okay, to of course to release the oxygen to our cells and to release the carbon dioxide from the cells to back to the lungs. But if carbon monoxide is there, okay, your oxygen and carbon dioxide can no longer be carried by your hemoglobin because your carbon monoxide binds very tightly to your hemoglobin. So your hemoglobin can be measured through this particular method. By the way, this is the reference method for measuring your hemoglobin. This is called Cyan Methemoglobin. These can measure all types of hemoglobin except for sulf hemoglobin. And how do we do this particular procedure? We need to have first a lysing agent to free the hemoglobin from the red blood cells. So we need to lyse our RBCs here. And then after lysing the RBCs, um, one particular reagent is being mixed with your sample, okay, so that your free hemoglobin will combine with your potassium ferric cyanide, and then your it will convert your hemoglobin to ferric form, your from ferrous to ferric, and it will become methemoglobin. So methemoglobin combines with the next compound within that reagent. That compound is called potassium cyanide to form a stable pigment called cyan methemoglobin. And the cyan methemoglobin now is going to be measured or quantified, okay, through spectrophotometer, okay, at 540 nanometer. So again, this is just a shortcut or a short explanation of the procedure. So when your RBCs are already lysed, the free hemoglobin, which has a ferrous form of iron, will be um, mixed with potassium ferric cyanide. So your hemoglobin will become methemoglobin after adding or after mixing potassium ferric cyanide. And then your methemoglobin will be mixed with another compound in your reagent, and that compound is called potassium cyanide. So thus your methemoglobin becomes cyan methemoglobin, and the cyan methemoglobin is the final product that will be measured at 540 nanometer, okay, in your spectrophotometer instrument. So now we will discuss about iron metabolism. Now, the primary function of our iron is to bind with oxygen and transport them to the different parts of the body. So iron is more stable in ferric state, and most of our iron in ferric state are bound to transferrin and ferritin. So in order for the ferric form of iron to be reduced again to ferrous state, it requires NADPH and a number of enzymatic reactions. So most functional iron in humans is in the form of hemoglobin and myoglobin. And usually the iron for hemoglobin and myoglobin are in ferrous form of iron. And the ferrous form of iron is incorporated into the protoporphyrin 9, which is found in our heme or hemoglobin. So by the way, protoporphyrin 9 is the precursor of your heme, and it is composed, or it is characterized by a ring of carbon, hydrogen, and nitrogen. So 1/4 of iron is in storage form, and they are usually non-heme iron or in ferric form, and they are stored in ferritin or hemosiderin um, in the liver or in the hepatocytes or in the macrophages. Small amount of our iron is also bound to our transferrin. So what are the forms of iron in the body? So number one, the storage form of iron as ferritin or hemosiderin in hepatocytes and macrophages. Second, those small amounts that are bound to transferrin, and um, the most part of them are present in our hemoglobin and myoglobin, which are the heme iron or the ferrous form of iron. So iron basically is a carrier of electrons. It is a catalyst for oxidation, hydroxylation, and other crucial metabolic processes due to its ability to cycle reversibly between ferrous and ferric states. It also must be regulated carefully because of its free forms. Okay? It plays a key role in the formation of oxygen radicals that can damage our cellular structure. I think you have heard of those free radicals. So if you have the free forms of electrons, okay, those can actually damage our cellular structures, okay, like your hemoglobin of your RBCs. So iron exists as a free cation transiently, and mostly they are bound or incorporated into various proteins. So having a free electron in the ferric form of iron, the oxidized one, it can form free radicals. Okay, and that can be harmful to our cells, like your RBCs. Whereas if we have ferrous form, okay, the electrons there are paired. Okay, so of course it's an even number, number 2. So the electrons are paired with ferrous form of iron. But with ferric, there is a free electron, and that free electron can form free radicals that can damage our cells. So that's why it's very popular right now that we need to take antioxidants to get rid of free radicals. So basically, what antioxidant would do is to donate electron, okay, to those unpaired electron. Okay, so those free radical has unpaired electron, and if you take antioxidant, it will help to pair, or it will donate electron to those unpaired electron in that free radical. So iron intake and loss should be balanced. Okay, so iron deficiency, that is when you have less iron in the body. Okay, it occurs from inadequate intake or excessive loss from bleeding. So when you don't eat much of food with iron, or if you experience bleeding, okay, or blood loss, that in that way you will have iron deficiency. So red blood cells starts to decline, its function starts to decline, and there will be decreased hemoglobin. Okay, of course, hemoglobin cannot function well without iron, right? So, however, if there is iron overload, which means you have a lot of iron in the body that probably hasn't been released, okay, outside. That's why there's a lot of um, iron building up in the body. So it could be caused by increased absorption or caused by repeated blood transfusions. And this overloading of iron in the body may cause iron toxicity that may damage our iron, not iron, our organ, and can lead to death. So iron status depends on our intake, our iron bioavailability, and our iron losses. So dietary iron, bioavailability of iron depends on its chemical form, whether inorganic or organic, or whether it is non-heme or heme iron. Okay, so it depends on the chemical form in the food and the presence of other food items that promote or inhibit absorption. So apart from intaking food rich in iron, we need also other food items that will promote and inhibit absorption of our iron in the body so that we can regulate our iron inside the body. So it is absorbed. Your iron can be absorbed in two forms, either heme iron or non-heme iron. The heme iron is the one with ferrous form. Okay, the iron that is incorporated in our hemoglobin molecule, and they can be absorbed more effectively. They are present in forms of our hemoglobin, our myoglobin, and heme enzymes. Okay, and then your non-heme iron, they are the ferric form of iron. They are inorganic. They are usually bound to transferrin and ferritin, and sometimes, okay, visible to developing erythroid cells. Um, and these are called Pappenheimer bodies. So the sources of our iron, food sources of our iron, for the heme iron, we can find them in the liver, meat, poultry, fish. The non-heme iron, we can find them in the leafy vegetables, legumes, beans, cereals, and milk. So iron absorption and excretion. So what influences iron absorption? Number one, it is influenced by the amount and type of iron from food. Okay, the amount meaning the quantity of food intake that is rich with iron, or the type, whether heme iron or non-heme iron. Okay, so second, it can be influenced by functional state of gastrointestinal mucosa. So it depends on how functional your gastrointestinal mucosa to absorb your iron, and also the current iron stores. Of course, the storage of iron in the body is primarily the ferritin. So it depends on um, the state of iron stores in the body, and erythropoietic needs. Okay, it depends on erythropoietic needs. So if we need to have more RBCs, of course we need also more iron. Okay, so again, it depends on the amount and type of iron from the food, the functionality of your gastrointestinal mucosa to absorb your iron, the current iron stores, and the erythropoietic needs. So iron absorption and excretion. So maximal absorption happens in the duodenum and upper jejunum of our intestines. Okay, so find where is duodenum in the picture and jejunum. So that those are the parts in the body where absorption of iron happens. So to be absorbed, your iron must be in heme form, okay, from the food, or converted from the ferric form to ferrous form. Okay, so conversion of ferric form is done by DCYTB, or the duodenum specific cytochrome B-like protein. Okay, heme now, the heme iron, the ferrous form, binds to the enterocyte in the intestinal absorptive cells in the mucosal epithelium and is internalized. So as you may see here, the ferric is being converted to ferrous by DCYTB, and with the help of DMT, your ferrous will go inside your enterocyte and be internalized. So your ferrous iron is transported by, again, your DMT1, okay, across your duodenal epithelium. Your ferrous iron is carried to the basolateral membrane, okay, base and sides of your membrane, and exported to the portal circulation and mediated by ferroportin, a basolateral transport protein. So ferroportin works with your hepastine. By the way, hepastine is a copper-containing iron oxidase, which facilitates reoxidation of ferrous to ferric. So once it is already ferric, it will bind to transferrin to be transported in the circulation. And some absorbed iron remains in the enterocyte as ferritin, okay, the storage form of iron. So what's the difference between ferritin and hemosiderin? Your ferritin is the main storage form of iron in the body, and its component is released by reticuloendothelial system, and this is a reflection of tissue stores of iron. It is a labile form of iron, okay, meaning the iron can get in and out of this form. So the form of iron of your ferritin is ferric, so it can be ferrous later. It can become ferrous, or it can go back to its ferric form. So it's really called a storage form because we can also use in times of iron deficiency in the body. Ferritin can be used. Okay, so this ferritin, okay, the ferric there in the ferritin will be reduced, or will be changed, or will become ferrous in order for the body to use them. Okay, so by the way, ferritin is also an acute phase protein, which means that it goes up in certain conditions like chronic inflammation. That's why when the patient has chronic inflammation, okay, your ferritin will increase. So in the picture, you have here a slide from a bronchoalveolar lavage presenting alveolar macrophages stained with hemosiderin. Your hemosiderin consists of ferritin and cell debris, and it is stable form, and that means it cannot be used, okay, because it cannot revert to the ferric in the hemosiderin cannot revert to ferrous, which is the heme form of iron. Now, hepsidine. Hepsidine is an antimicrobial peptide produced by the liver, and it acts as negative regulator of intestinal iron absorption. It also binds to your ferroportin receptor, causing degradation of ferroportin. So basically, hepsidine is actually a regulatory peptide. Okay, and it results in trapping of iron in the intestinal cells. So this particular peptide, okay, helps the body to regulate iron absorption. So hepsidine is also an acute phase reactant, and it is also a hormone that is produced by hepatocytes to regulate body iron levels. So it helps in the absorption of your iron in the intestine. It regulates the absorption of iron in the intestine. And yes, it regulates the absorption and release of iron also in the macrophages. So hepcidine production is directly proportional to our body iron. So when your iron increases, it will also increase. When your iron decreases, it will also decrease. So hepcidine, what the hepcidine does, it reduces iron absorption in the enterocyte, and also it increased iron accumulation in the macrophage. So what are the sources of our iron? It is from our diet, okay, from iron recycled from the extravascular hemolysis. So summary of iron absorption. So your heme iron, your ferrous form, and your non-heme iron or your ferric form. So of course, your heme iron can be readily absorbed, but your ferric form must be um, converted to a heme form. So who does it? So DCYTB is the one that converts your ferric to ferrous form so that it can be absorbed. Now, in order for the ferrous form to go inside the enterocyte, it will be facilitated by the DMT1. Okay, it will go inside your enterocyte or your intestinal absorptive cell, and with the help of your ferroportin, okay, and in conjunction with your hepastine, okay, your ferrous form will become ferric form to be transported in the circulation by your transferrin. Okay, so the ferric form now, okay, will be carried by the transferrin to be delivered in the bone marrow. So hepcidine again is present in order to regulate, okay, the absorption or the release of your iron in the enterocyte. So iron cycle and trans. So now transferrin is with iron already. Okay, so usually transferrin carries the iron to your hepatopoietic tissues, okay, which is found in your bone marrow. So in the bone marrow, your transferrin will bind to your transferrin receptor on the cell membrane, and the transferrin receptors are bound to erythroid progenitors and precursors that are rapidly dividing, except for our mature RBCs. So transferrin is taken into the cell by endocytosis, and there will be endosome formation, and the iron will be released from the transferrin by acidification of the endosome to pH 5.5, and your iron transported across your endosome membrane DMT1. So humans regulate iron by controlling absorption too. So normally, okay, we tend to absorb iron 1 to 2 milligrams per day. So if currently you have decreased iron stores in your body, okay, chances are your body needs more iron. So that's why the body wants to absorb more. So if you have less iron stores at this point in time, okay, your body will absorb 3 to 4 milligrams per day. But if your iron is overloading inside your body, okay, your body will also absorb less. Okay, so it will absorb 0.5 milligrams per day if you have iron overload. So ferrokinetics, by the way, ferrokinetics is the movement of your iron from the body starting from the absorption of iron. So it includes the following. So the transferrin, it includes your transferrin receptor and your ferritin. So transferrin is produced by our liver, your liver cells, or your hepatocytes. The major function is to transport your ferric form of iron from the plasma to the normoblast in your bone marrow. And transferrin binds to your transferrin receptors on the normoblast membrane. And the half-life of your transferrin is 8 days. Transferrin receptor can bind two molecules of transferrin. Transferrin to transferrin receptor affinity depends on the content of iron and its pH. Okay, the highest affinity, okay, happens with diferric transferrin in the pH of 7.4, and apoferritin, by the way, is a protein component of ferritin without iron. So apoferritin, no iron within the ferritin. So your transferrin receptors can bind to ferric iron. Okay, so this one is called diferric transferrin. It has two ferric iron in it, and the absorption of this is through endocytosis, and it will form endosomes, and the pH 5.0 triggers the release of those ferric iron from the transferrin. And when the transferrin has already been detached with the ferric iron, that transferrin will be called apotransferrin, okay, which means no iron in it. Okay, so what triggers again for the ferric form of iron to be attached with your transferrin? Okay, it is when the pH is 7.4. So for the laboratory assessment of your iron stores, so laboratory assessment to measure our iron levels. Number one, serum iron concentration. It refers to the ferric bound to our transferrin. It exhibits the diurnal variation because usually it is highest in the morning and lowest in the evening. And your serum iron concentration, when the patient has iron deficiency anemia and inflammatory disorders, their iron concentration, serum iron concentration, are decreased. Next, test for iron uh, measurement is your TIBC, or your total iron binding capacity. This refers to the total number of available sites of transferrin. And then next is our percent transferrin saturation. This is the percent of transferrin carrying iron. Next is the serum ferritin levels. This is the levels of stored iron, which declines early in uh, iron deficiency anemia, or IDA. The free erythrocyte protoporphyrin may also be seen in excess when there is IDA, because remember that um, if you have um, excess protoporphyrin, that means you have less iron to pair with your protoporphyrin. So it it shows that the patient may have deficiency of iron in the body, because how come there are free iron protoporphyrin present? Okay, no partner. Okay, it should be the iron that will partner your protoporphyrin. And then another test is your Prussian blue staining. It is an iron staining, and this will exhibit iron concentration in your macrophages, in your nucleated RBCs, and in the reticulocytes. So the terms for those RBCs that are not yet matured, either nucleated or reticulocytes, that has iron content with it is called sideroblast. But for those RBC, the mature RBC that has iron content in it, you call that siderocytes. So again, sideroblasts are those erythroid cells that are with iron stores in it, and your siderocytes are RBCs that has iron content in it. Okay, so in this picture, it is an example of a smear being placed under the microscope to view the erythroids. Okay, and this particular smear has been stained with iron staining, your Prussian blue, and those pointed cells, okay, are actually erythroblasts. They are um, immature RBCs that are with iron. Those blue colored stains in it are the iron content. So this is the end of our discussion for this topic. Thank you so much.